Good morning. As we start this next big topic, I want to remind all of you students not to fall behind. After you've listened to videos or gone to other lectures, make sure you go back and review those notes within 48 hours so you don't forget any information and then review it again the next week. This is how you keep up to date on material. You don't have to start again at ground zero when studying for tests and quizzes. Make sure for this course that you're drawing and labeling pictures because it's very picture heavy. And when we have pathways, practice those pathways over and over again and go to the Learning Center and make a study group. So now we are ready for a new topic. We're going to learn all about cells, and we're going to start in this first section just with some basics and some general information about cells. So here are some pictures of cells. You may recognize from high school, having looked at cells, an amoeba or a paramecium or a plant cell, even a sperm cell here. Cells were first seen in microscope in the mid 1600s, mid to late 1600s. I think the official date was something like 1674. And since then, we have learned a lot. We know that nothing smaller than a cell is alive. So if you go back to that hierarchy of life, you know, where we started with the atom and moved on, everything smaller than a cell, none of those have the properties of life. Okay. So the cell is the smallest thing that contains all the properties of life. So what that means is all the processes of life must take place within a cell. And a cell can be an entire organism, or an organism can be made up of multiple cells, like a human body. And so lots of experiments have been done with cells, lots of hypotheses, and over the years they've been validated. And so we have developed what is known as the cell theory. And there's three components to the cell theory. The first part is the cell is the basic unit of life because nothing smaller is alive. That makes it the most basic thing. Second, everything that's alive is made up of cells. If it doesn't contain cells, it's not alive. And third, new cells can only come from pre-existing cells. Now, there's different methods for that, and we're going to learn a couple of different methods for making cells in the human body. But first, I want to take a little diversion and tell you about a really important person when it comes to learning about cellular biology. And that person is Henrietta Lacks. And her legacy are what are called HeLa cells. Because when we name cells, they are named for the person for whom they came. The first two letters of the first name followed by the first two letters of the last name. So Henrietta Lacks gave us HeLa cells. And Henrietta Lacks was a very poor tobacco farmer from Southern Virginia. And after she got married, she moved with her husband to Turner Station, which was a very historical Black community outside of Baltimore. And they had multiple children. And when she was raising, shortly after she had her fifth child, she went to John Hopkins Hospital in Baltimore, and doctors discovered a tumor on her cervix, the lower part of her uterus, and biopsied and saved a sample of her cancer cells in that biopsy. And this was standard practice. There was no informed consent. Can we do anything with those cells? And that was standard practice in those days. And it didn't matter if you were a rich person or a poor person. Informed consent did not exist back in those days. Now, as far as what was done, public poor wards there was a lot more done without the patient being aware of. And that didn't matter if you're black or white. The difference being in public wards, you would have 20, 30 patients sitting in a room as opposed to private wards. Anyway, she was quite young when she died. She was only 31 and left 5 young children. And she was buried in an unmarked grave because, like I said, the family was very poor and they didn't have money. So at that time, most of the scientists, when they would do biopsies, the cells would die shortly thereafter. And her cells actually were the first cells that survived. And in fact, her cells thrived in laboratory. And this is what made them so unique. And so they became known as the first immortal human cell line, as in scientists have been able to culture them indefinitely since 1951, meaning researchers around the world have been able to reproduce these studies using identical cells. So I want you to go back to this picture and look at this cell right here, this round cell with this very small little nucleus. This is a cell as it would look if you took it during a normal pap smear, ladies. This is what a normal cervical cell looks like. Whereas these are Henrietta cells, cervical cells. So as you can see, these cells look angry or nasty in comparison. Like this cell here has 2 nuclei with a bunch of nucleoli. Look how huge this nucleus is in comparison to the size of the cell. This cell is undergoing rapid mitosis going on. These cells have multiple projections. They are going rapid growth, and none of these characteristics are normal for cervical cells. Anyway, these HeLa cells are the workhorse of cancer research, AIDS research, drug research. They've flown into cells. They were used to develop Jonas's soft polio vaccine in the 1950s. They've been used for Parkinson's research. They've been used to develop flu vaccines, to map genes for cloning. They've been used to look at the effects of atomic bombs, et cetera, et cetera, et cetera. Oh, and by the way, they do contain HPV. So all of you male and females, make sure you get your HPV vaccines. Okay. But all of this, I want you to realize that Henrietta Lacks is the most important woman in the history of medicine. And she is a Black woman. And here's what's very interesting about her cell line. About 10, 15 years ago, a bunch of research was done looking at the other cell lines that had been developed since 1950s. And what was realized is about 20% of those cell lines had been contaminated with HeLa cells. And in fact, they were not pure cell lines because these cells are practically indestructible. So yeah, it's impossible to kill them. So this is a $216 billion industry, her cell line. And her family, her descendants only found out this story in the 1970s. As I said, there was no informed consent back then. And so there was a very famous book called The Immortal Cells of Henrietta Lacks, which was made in 2010. And Oprah made it into a movie in 2017. And one of Henrietta's grandsons made a memoir, a family memoir that was released around the same time. And the family sued in 2020, 2021. What was interesting was the family actually did gain some control around 2013 as to what her cells could be used for. But you know, this is 50 plus years after use. So they didn't gain that much control, but they were able to limit a little bit of what the cells are used for. Their lawsuit against the Fisher Scientific Company, which actually controls their cells and makes money off of selling their cell line. And by the way, if you don't think this cell line brings Fisher Scientific money, it's been used for more than 75,000 studies and more than 110,000 scientific publications. So a settlement was made with the family, and it's a settlement that is private, but it's estimated to be at least $12 million to the family, and more likely probably closer to about $26 million that the family got in compensation. So that's just an interesting tidbit about cells. But let's get back to what it is you need to know. So how do we look at cells? Well, here's the thing. You can't see cells by the naked eye. I know when you look at your skin and you look up close and you can see like little triangular or pentagon shaped things, everybody thinks, oh, look, I'm looking at cells. You're not. Okay? In order to see cells, you have to look under a microscope. And the first microscopes were invented around the mid 1600s. And so here's a very early microscope from 1665. And here's a modern microscope, better than the ones you probably had in grade school, more akin to one that we have at our college. And the ones that we have all cost about $1,000 each. And here's what they have in common. Up here at the top is your eyepiece. Earlier microscope, that's all it was, it's the eyepiece. Now we have a magnifier, a 10x magnifier up there. There was another magnifying glass down here. We can rotate a 4x, a 10x, and a 100x magnifier down there. And then down here, you have a piece of glass where you're looking at the specimen here. We call that a slide. You can look at a specimen. Now, that specimen doesn't have to be dead for a light microscope. So you can put a drop of pond water, for instance, which is teeming with microorganisms, and look at it. And then what you have to have is a light source. So back in the day, in the 1600s, 1700s, 1800s, they would have a candle or some other light source here on the side, and then some sort of mirror to try to concentrate the light over here so they could see. Nowadays, our light source is underneath, so the light is going through what it is we are looking at. And so here's an example of what you would see in a light microscope today. And so light microscopes allow us to magnify things from a low of about 4 times magnification, about the same thing you can do with a magnifying glass, to a maximum of 1,000 times magnification. If we want more than that, we have to use what is called a transmission electron microscope, and these were only developed in the 1930s. For this, we have to take a super thin slice of the material, much, much thinner than a piece of paper, thinner than a strand of thin hair. Okay. And then on that strand, that thin slice of whatever, it gets coated with metal and gets put in a vacuum chamber through which electron stream is run. And then you view that on a monitor, and you would get a very crisp, clean picture in black and white that looks like this. And then, yes, you can colorize it just like a color TV. So you can make this part look pink or red or green or whatever you want it to look like it, to bring out contrast between things. And this is what allows us to look at details inside cells much more clearly. I think you clearly can see details, like this is looking at the inside of the nucleus. This part right here is looking inside the nucleus of a cell. And this right here is a totally different cell. Now, the problem with this is it doesn't let you look at interactions between cells and look at outside surfaces of cells very much. And for that, we look to a third type of microscope known as the scanning electron microscope, which was developed in the early 1960s. And in this time, once again, we coat this substance with metal, put it in a vacuum tube, and stream electrons over it. And here's one showing that it's been colorized. And here we have these cells with red, and then here's a different kind of cell, just an orange color. And then there's this other substance, which is this yellowish green or this greenish yellow color. Now, what I want you to realize is this cell right here in the light microscope is the exact same cell as this barbell-shaped cell right here in the transmission electron microscope, which is the exact same cell as this red cell right here in the scanning electron microscope picture. Do you have any clue what cell that might be? That's what one of your red blood cells look like. And you see how we can get much different information about it. So we'll come back and learn a lot more about the red blood cell later on when we learn about the cardiovascular system. And you'll learn why this red blood cell is so skinny. And why, when I was saying, look, here's the nucleus of a cell, why we're not seeing anything that looks like the nucleus of a cell here. Okay, so moving on. When we look at the size of the structures, okay, so this is science. We do everything in the metric system. Here's the part of the stuff that we can see, you know, when we talk about science, we go from the electrons all the way up to galaxies. Let's talk about stuff we can see with our naked eye. Huge from the whale, we can see that. We get smaller to a mouse. All right, so here is a centimeter. So an inch is just about 2.5 centimeters. So this is a little bit more than a third. Sorry, a centimeter is a little bit more than a third of an inch. Sorry, but less than half of an inch. Okay, and a centimeter is made out of 10 millimeters. So a fruit fly is a little bit bigger than a millimeter. It's closer to 2 to 3 millimeters in size. Okay. And then we can clearly see that we can see all the way down to the size of a single hair. Notice we cannot see the size of a cell. So here's what we can see with the naked eye. But with a light microscope, we can cross over with the naked eye. We can see a cell. We can see some stuff inside the cell. We can see bacteria, but notice we can't see anything really smaller than bacteria. And in fact, we can't see some of our really tiny bacteria. In order to see a virus, we have to use an electron microscope. We also have to use an electron microscope to look at structures of proteins as well as our other smaller molecules. All right. So now let's look at our cell sizes and our shapes. Here are 3 different cells where the outer, and they're all squares, but the dimension of the square is 1 millimeter versus 2 millimeter versus 3 millimeters. And all 3 of these have the exact same size nucleus. So what you're realizing is the volume of the cell is increasing much more rapidly than the surface area. And here's the problem with that. Stuff can only enter the cell through the surface. Waste can only exit the cell through the surface. So a cell to grow perfectly as a square or perfectly as a circle, because like didn't most of you learn in grade school, middle school to draw a cell as a circle? That's bad. Okay. Cells don't like to be perfect squares and perfect circles because they like to have at least one relationship to be smaller because that's much more efficient for the cell. If a cell is square like this with the height and the width and the depth, then something in the middle's got a long way to travel. But if it's narrow like this, it only has a short distance to travel. Like, look at your cell phone. It would have a huge distance to travel if this was the smallest dimension. It likes this really short dimension, this really short distance. That's ideal for cells. All right. Now let's talk about types of cells. So I've alluded to this before. There's two types of cells. Prokaryotes are the types of cells that do not have a nucleus because pro means before, kary means nucleus. Yes, they still have genetic information. Yes, it's still going to have nucleic acid. It's just not going to be contained inside a nucleus. So an example of a prokaryote, there's a drawing right there. It has an area containing the nucleic acid called a nucleoid region, as opposed to one with a true nucleus. Just take the UE of true, switch those letters around, and you have eukaryote. Right here, it has a true nucleus. Yeah, notice both of these have plasma membranes. Both of these have cytoplasm containing organelles, such as ribosomes and some other stuff going on. Both of these can have plasma membranes, yes. Okay. So looking at a prokaryote, these are all going to be one-cell organisms. So your bacteria, streptococcus, staph, gonorrhea, syphilis, E. coli, Clostridium, all the wonderful thermophiles that you would see at hot springs giving you all the beautiful different colors. So bacteria, archaea, tons of different types. Those are all going to be prokaryotes. Eukaryotes going to be larger, more complex. Yes, some can be unicellular, but we're going to be looking at at multicellular organisms, humans. Okay, we have eukaryotic cells. Okay, so yeah, from now on, we're going to be learning about the human. We have a plasma membrane. This is not the same thing as a cell wall. I'll talk about a cell wall. I think I have the next slide. I show you what the cell wall is. Okay. Eukaryotes have a plasma membrane, and we have some organelles, many of which have membranes in them. And our DNA, by definition, because it is a eukaryotic cell, is found within a nucleus. So there's two types of eukaryotic cells. There are animal cells, that's what we are, and there's plant cells, that's what we are not. So two huge differences. Both are going to have plasma membranes, those cell membranes, but plant cells outside the cell membrane have a rigid cell wall. And so what happens is inside the plant cell, they have a special type of organelle called a central vacuole. It's similar to it's a specialized type of like vesicle, which can store stuff. We have vesicles that can store stuff, like, for instance, we have vesicles that can store adipose, that store lipids in our adipose tissue, in our fat, okay? They have a vesicle called a central vacuole that stores water. So if the plant cell is in a very watery environment, such as if you take a stalk of celery, you stick it in a glass of water, that central vacuole will fill up with water, and that puts pressure on the cell wall, and then you have a crisp piece of celery. Now, if you were to take that piece of celery and put it in salt water, what would happen is the water would leave that central vacuole and would go and try to dilute out the salt water, and that piece of celery would become limp. Okay, so those are the two big differences between the animal cells and the plant cells. Okay, so last thing I want to do is I want to give you a prompt for active learning. Do you remember when you learned about the 3 different domains and the 6 different kingdoms? Go back and write them down. And now, after you've written them down, write down whether each kingdom is prokaryotic or eukaryotic. Okay, and that'll be a good review for you. And with that, we are done with this section, and I will see you shortly when we come back and we start learning about cells and details. Thank you.